And it's recording. And we're live. In 3, 2, 1. Welcome to the Unbelieving Scum YouTube channel. I'm Dr. Z, your host, the Unbelieving Scum, right here. And this is episode 17, first installment of How Science Works. What we're going to talk about today is 60 years of advances leading to the COVID mRNA vaccines. A phenomenal story about science. And yes, Virginia, there is a method to science, and all PhD researchers around the world are trained in it, despite what some philosophers try and tell you, that the scientific method doesn't exist, or it's a grab bag of different things, and this is bullshit. Okay, here's what the scientific method is. It starts off with systematic observation and analysis. You study the crap out of whatever it is you think you're going to try and explore, investigate, discover, and talk about. Then you do theory, hypothesis, experimentation. Your work's got to be replicated, and it's got to be published in peer-reviewed science journals. All scientists do this method. Okay, again, I don't care what the philosophers say. We're going to concentrate on just number one, systematic observation and analysis today. We'll talk about the whole thing at some other time, but you really have to study the crap out of whatever you think you're going to work on. Okay, if you're a PhD researcher in any field, you're going to get a 4 years bachelor's in that field. Then you're going to focus more when you get your master's, another 2-3 years, write a thesis. And then you'll focus even more or replicate what you were doing in your master's with your PhD research, another 2-3 years, okay? And when you get done with that, you're probably, in most fields, get a postdoc and work in somebody else's lab for a year or two, and you're damn near 30 years old before you're applying for professorships and full-time research positions. And you're going to spend the rest of your 30, 40-year career working on whatever you were into. That is how it works, and we all pretty much follow that same path. So, science is cumulative, okay? It's not revolutionary, as some philosophers claim. It's not haphazard or clutching at straws, such as usually wrong, as some Christians want you to always believe, nor constantly overturning, rewriting, upending previous theory or explanations, as a lot of journalists want you to believe with their sensationalist headlines. Okay? The COVID-19 vaccine development is a great example of typical science. Okay? 60 years of discovery and advances that led us to having mRNA vaccines mass-produced in 2021 and worldwide. It's phenomenal. Systematic observation in excruciating detail of not just mRNA, but gene therapy techniques, and the understanding of AIDS and various coronaviruses. If we hadn't done all that in the past 60 years, no way we would have had those vaccines within a year. Okay? There's 3 key areas of advancement in this. Number one, mRNA discovery and synthesis, okay? Gene therapy techniques, okay? And AIDS vaccine R&D and production distribution setup. Those 3 areas are where most of these advances are going to fall in, okay? And all of this developed independently in dozens of labs around the world involving hundreds of researchers, just figuring out how things work in each area. Okay? There was no top-down, central organized, single operation 30 years ago or whatever dedicated to, hey, let's make an mRNA vaccine by 2021. You know, that's not how it went down. Science and innovation of any kind develops often, most often, in wholly separate and unforeseeable steps, which can converge at later times, okay, to spawn a whole host of creative and fruitful advances. The light bulb's an example, lasers, the automobile, and we'll probably talk about the light bulb and lasers. The time was ripe for someone, often more than a few, to assemble all these advances into the next big innovation. Matt Ridley, and I'll give you some links here, has really explicated this in a couple of books and some talks, okay? So, for example, nobody said, hey, Einstein just described lasers in 1915, and now we can put barcodes on every product imaginable, and Pink Floyd can do killer light shows. Wow! Yeah, 100 years later, all that happened. Nobody said that when 1915 and Einstein's laser paper came out. When the first transistor was invented, right? Hey, now we can have teenagers listen to rock and roll at the beach with a portable handheld radio. Bitchin', dude, said nobody ever. Okay? It's not how it went. Okay? It's not how it works, and I'll do episodes on laser and transistors someday down the road, like I said, maybe the light bulb too. And the mRNA story is no different. An incredible story of accumulated knowledge, discovery, technology, and organization, and it happened worldwide. So here's the timeline. 20 advances over 60 years that contributed to the almost immediate development within a year and mass production and distribution of mRNA vaccines. So we're going to take them five at a time here. Okay. 1960, mRNA is identified. Okay, if those of you remember, 1953, DNA was first figured out, discovered, described. Took another 7 years to figure out there was messenger RNA in there, right? 1960 to 1984, quarter of a century almost, 24 years later before we could synthesize and make a little strand of mRNA, right? Next big step, 2002. Almost another 20 years later, the first SARS outbreak, the first sudden acute respiratory syndrome, happened in China, worldwide SARS-CoV-1 virus from bats through civets. That happened, okay? Wasn't as bad as our SARS, right? But it was important that it happened. A lot of people were geneticists, virologists, immunologists. It really perked their ears up. They were prepared. What are we going to do with this kind of stuff? By 2004, next big advance, mRNA is coated in a lipid package to survive bloodstream injection. That's a key move forward. We are trying to develop gene therapy techniques, but these tiny little strands of DNA, mRNA, whatever, they won't hold up through injected into your bloodstream. So we had to figure out how to coat them in tiny balls of fats to survive being, vaccinated, bloodstream injection. 2005, the first modified mRNA successfully worked in mouse cells developed by another lab. So they started to figure out how to do it, make it work, and as usual, we try things out in mice first. By 2008, the RSV, the respiratory syncytial virus vaccine, forget what it is, was based on protein structures, right? We came up with a vaccine for them based on figuring out the proteins that we could get your body to recognize and attack this RSV. Next big step, 2012, MERS came around. Middle East respiratory syndrome, CoV 2012, came through camels to humans, another zoonotic transfer, typical of viruses, all kinds of diseases. Again, in the coronavirus family that's probably from bats, 35% of the people that were infected died. I mean, that's an insane number. 30 of the people infected died. That's huge. Luckily, it never got out of the Middle East. It wasn't as infectious as it could have been, versus 0.04% of people die from COVID, okay? But it's highly infectious. So it's not as deadly, but when you have hundreds of millions of people infected worldwide in a short time, 0.04% comes out to a big number, which was about 7 million people died worldwide. Okay, and then you have long COVID complications that we're just still figuring out. Okay, 2013, we had a new approach using a coronavirus, common cold virus, in order to develop a defense against MERS. They kind of changed their attack. Instead of attacking the virus directly, they said, look, let's figure out some of the simpler coronaviruses that cause common colds, things like that, and see if we can figure out how to attack those. We can then attack MERS or SARS or whatever. 2015, Bill Gates did a TED Talk, not on his prescient ideas, but he's talking about what the scientific community was worried about, that there was an eventual global pandemic coming at some point from some kind of virus someday, like SARS, MERS. And so far, SARS and MERS were about 10 years apart. We could have one in another decade, right? Not crazy at all. It wasn't, telling the future or anything. It was just, hey, we got to watch out for this. Here's what we've seen thus far, right? 2017, the common cold coronavirus coronavirus spike protein was synthesized. Again, a quote from Richard Feynman, I can only understand what I can build, or I can only build what I understand, whatever the exact quote is. The point is the fact you have to learn to assemble it, synthesize it, make it. Then you really understand how it works and how its structure is. So we figured out how to make a spike protein off a common cold coronavirus. Huge step forward. Number 11 is not a good one. 2018, that idiot Trump dismantles the National Security Council Pandemic Unit. We would have been much more prepared, much smoother distribution, everything else, if we had that group that was purposely put together for the coming type of virus that was going to happen, and they were dismantled 2 years, less than a year, about a year before COVID-19 showed up. December of 2019, COVID-19, the pneumonia-type virus, the disease breaks out in China late in the year, December. Within a month, 2020, the SARS-CoV-2 virus was sequenced. All 30,000 little base pairs in there. Phenomenal work. The fact that we could do this kind of stuff. If this was 5 years earlier, we would have been screwed. Sequencing would not have been that quick. Okay? 2020 February, month later, the spike protein structure is published. Here's an image of what it looks like. By 2020, in March, Fauci here in the United States initiates the clinical trials of a SARS-CoV-2 mRNA vaccine. Again, they had everything all set up to do AIDS vaccines. We learned how to synthesize mRNA a decade or more before, so around the world we could put together, the big pharmaceutical companies put together an mRNA vaccine. 3-4 of them did it within a couple months. 2020 in March, 30,000 volunteers recruited to enroll 2,000 people per day to participate in clinical trials for 4 vaccines across 100 sites. Okay, this is the United States. 2020, April, October, 100,000 people participated in the clinical trials, right? 2020, by November, the results of the clinical trials came out, and we're showing 95% efficacy in keeping people out of the hospital or dying from COVID-19. Phenomenal success rate. The researchers were thrilled. We pulled this off in about a year, and by 2021 January, the big once Biden was president, the rollout of mRNA vaccines. Trump was going to get 20 million vaccines out before he left office, but he was too busy fomenting an insurrection to bother with that kind of mundane stuff. So anyways, by 2021, we were vaccinating the first responders, elderly, general population, worldwide, January, February, March. Okay? So, pretty phenomenal 20 steps, right? And it wasn't just the U.S., it was a worldwide rollout. In fact, England beat us by a week getting the vaccines out. Now, one thing Trump didn't fuck up was the combined public-private effort, Operation Warp Speed, remember? That worked very well in the U.S., but like I said, we were doing what everybody else was doing. England beat us by a week. To give you an idea of the scale we're dealing with here, the period at the end of a sentence on your typical piece of paper you read is about 0.5 millimeters. 10,000 COVID viruses or 10,000 mRNA molecules in the vaccines both could fit inside that little period, that tiny dot. 10,000 mRNA molecules or 10,000 viruses are the size that can fit inside a period at the end of a sentence. Phenomenal. They're measured in billionths of a meter, nanometers. They're on the order of about 100 nanometers. For comparison, the wavelength you see of light that appears to you as red is around 700 nanometers, right? 7 times bigger that wavelength of light is than the actual coronavirus or an mRNA molecule. Okay? Just phenomenal. The fact that we can identify and sequence the 30,000 nucleotide bases, the letters that make up the single strand of RNA in a tiny SARS coronavirus, the fact that we can, see it with our microscopic methods, sequence it with DNA sequencing methods. You kidding? 10,000 of them could fit in the period at the end of the sentence, and we can sequence all 30,000 letters in one of those viruses. Incredible. Okay. It should give you awe, but as far as I'm concerned. And we give you a tiny mRNA strand in the vaccine that triggers your immune system to react to those proteins on the spikes of the coronavirus, right? And we coat them with a nanoparticle-sized lipid fatty coatings to keep them intact so they get into your cells, tell your cells to make that protein. The protein goes out in your bloodstream, your immune system recognizes it, and now your immune system has antibodies against the spike protein should you get infected by COVID-19. You're ready to fight it. And it gives you 95% protection against being super sick or winding up dead or in the hospital, right? Almost 7 million people died worldwide from SARS. Over 1 million in the U.S. alone out of 676 million infected worldwide, which is about 1%, okay? Infection rate was around 3, okay? 3 more people infected on average by each infection person. That's why it expanded exponentially. Exponentially? I can't talk today. Exponentially. It's not as deadly as other viruses, but it was incredibly infectious. So 1 person infects 3, 3 infect 9, 3 people, each of those 9, 27, you get it? Exponential? Okay. So it's incredible. Okay. By contrast, the 1918 Spanish influenza pandemic killed 50 million or more, not 7 million, 50 million worldwide, with 500 million infected, about a 10% death rate. No vaccines, nothing to protect it, keep you from getting super sick back then. We gave people worldwide a 95% chance of not being super sick, hospitalized, or dying with these mRNA vaccines, right? And we kept the deaths to a minimum, so now COVID is like an annual flu thing. It's endemic in the population. Most of us have either been vaccinated or had it at least once, so you're not going to see the pandemic you saw in the past. I had firsthand experience here in the U.S., duplicated in counties all over the U.S. I worked for my local county health department. I volunteered to help administer the vaccine. They needed people just to direct traffic. We would run 1,000 people through a rented out empty Kmart shopping center. We would set up and run 1,000 people through there in a Saturday afternoon. Half-day session, right? 15, 20 nurses all doing shots, observations. It was awesome to be a part of it. What a rollout. So this whole episode is but one classic example of how science works, okay? Dozens, maybe hundreds of advances allowed us to have mRNA vaccines or lasers, transistors, and a bazillion other devices that came with it, right? Religion got you none of this. In fact, most of the vax denial, right, are hardcore religious types. And I will have an episode on that one day too, for sure. But for now, ta-ta for now. Last thing I guess I want to say is just that this is how science works. Everybody operates by that scientific method that starts with incredible systematic observation, data collection, and analysis. You really got to know your subject. We've done the easy stuff. We know water boils at 212 and lead melts at this, and we know the structure of the water molecule. We're working on the hard stuff now, whether it's mRNA vaccines, figuring out how to cure Alzheimer's or how to stop autism or, what have you, climate change, right? We're working on the hard, big stuff now, and I'm confident we can do a lot with all those things. Just the advances in my lifetime have been fantastic. So anyways, ta-ta for now. If you like anything you see on this channel, please hit that subscribe button. Send me a comment. My Unbelieving Scum email is there. Hit the Patreon button if you can afford it, and try and support my work any way you can. I love doing this stuff. This is fantastic. And science education is where it's at. Information transfer is what separates us from all the other animals, and I'll have an episode on that someday too. So adios for now. Talk to you later. Have a good one. See you.